Okay, so good evening, everyone, and welcome to our second class on non-sterile product compounding. Um, I see at least three persons who were in the last class. I'd like us to start today's class with some form of recap. So I will ask persons who were in the last class to say one thing they remember from last class. I think it's important we recap, we know where we left off, and then we continue. Non-sterile product compounding. So, anyone remembers anything from last class? Just one thing per person, and then we move on. Anyone remembers anything? Rachel, you were in the last class. Poker, were you in the last class? Dr. Goodness, I also remember you in class.
Yes, sir.
One thing you remember from non-sterile product compounding from last class, anything before we continue?
We talk about preparation of customized medication not required to be sterilized and
Correct.
some key principles.
Absolutely correct. Very correct. Thank you. You did remember.
Thank you, sir.
That's right.
Yeah.
Yes. When we talked about patient-centered care and why
Yes.
compounding is important because they are
Yeah.
patient-specific. Yeah. Good.
Yeah.
Good one. Good. Rachel, remember anything from last class?
Yeah, we talked about the indications for non-sterile compounding, common dosage, common dosage forms,
Correct.
principles of patient-centered care.
Very correct. Very correct. There are about 8 principles. We talked about that. Patient's needs, preferences, and then respect for patients, communication, ensuring continuity of
Decision
care
making and
access.
interaction.
Correct. Correct. Very good.
Yeah.
Good. All right. I see Precious just joined. Uh, Dr. Goodness, you want to say anything, or should I continue? Okay, so let me just go ahead. I'll just do a run through of what we discussed last class. I see that we remembered a lot, which is quite encouraging and very good. We talked about non-sterile compounding in the context of patient-centered care, and we said is a model of healthcare delivery that places the patient's needs, values, preferences, and experiences at the core of decision making by the healthcare provider in the healthcare provision process. That's was something we talked about. We also went through 8 key principles of patient-centered care, and it was important for us to go through this because compounding generally, sterile or non-sterile, as we'll find out, is something that relates to patient-specific needs. And so we needed to lay that as a background last week. We also differentiated between sterile compounding and non-sterile compounding. We said sterile compounding is compounding of medicines or different ingredients to make specific medication for specific patients in a situation where germs is completely free of germs, the environment is free of germs or any microorganisms that are viable. Why we said non-sterile is compounding in a situation that not necessarily free of every germ and microorganisms. We differentiated that. And we also talked about reasons for non-sterile compounding or indications as we refer to them in the pharmaceutical space, is to say, you don't necessarily have to make it very sterile in terms of, going through all the efforts to ensure no germs, no microorganisms, because of the indications and route of administration. We're saying indication includes things like pediatrics needing liquid dosage forms for medications that are not readily available in liquid form. They're in tablets, but you need to prepare specifically for a child or a pediatric. Those are indications. Patients that are allergic to certain things that you find in a particular medicines. Maybe they're allergic to a preservative or a stabilizer within the preparation. So you need to use the same ingredients but exclude that particular preservative. We talked about customized doses, that are not commercially available and such. And also ended the class by saying that the common dosage forms, while that require non-sterile preparations, could be oral routes, when you do solutions, suspensions, and syrups that you take. We know we have the a defense system within the guts that can fight it. So you don't have to go to that length in making sure that everything is sterile before you prepare it. Topical preparations. Topical simply means skin. Medications that are applied on the skin, ointments, gels, lotions. You don't have to necessarily bother yourself to making sure the environment is very, very, very sterile for you to prepare them. I also talked about some rectal and vaginal medications, suppositories, and pessaries. That was where we ended in the last class. And so for today's class, as you see, we will continue with facilities and environment for non-sterile product compounding. That's we're going to be talking about facilities and environment. We will also be looking at apart from facility and environment, today we'll be looking at equipment and materials used in non-sterile product compounding. We will also look at compounding ingredients, and we'll end with a discussion on pharmacopoeia. And I did mention something about pharmacopoeia last week as the official document that contains standards for medicines and pharmaceutical substances. So we'll look a bit into that today. So 4 quick subtopics, and we're done with today's class. Like I said, facilities and environments, equipment and materials. Then we'll look at compounding ingredients themselves, and then we'll end with pharmacopoeia. So facilities and environment for non-sterile product compounding. The emphasis for us here is that environmental control is, for non-sterile product compounding, is as important as the sterile compounding. I'm saying that we will take the environment to be serious. We want it to be as enabling as possible. And we will not say because it's non-sterile, we will not make any effort to make our preparation very safe. And so I've just highlighted a few bullet points we'll go through to talk about facilities and environment. The first point being dedicated compounding area. A dedicated compounding area. I mean, for sterile, you already know that has to happen. For you to have an environment that is free of any germs, you have to have a secluded area where that preparation is happening. But we're saying that a dedicated area, like it doesn't have to be a room or a closure, even though in modern times, this is what we're beginning to see. In my days of pharmacy practice, in school, and even when I was practicing, we had a compounding section, and with just benches. And then for non-sterile compounding, you just have a bench that is clean and an area where you can work on. But these days, we're already beginning to see some kind of modular construction at the end of pharmacy sections to happen. So you have to have a dedicated area. We won't say because it's non-sterile, we'll just compound where in the place where we're doing dispensing or where we're storing commodities, we'll just go and compound them. No, you need a secluded area, an area that is dedicated to say this is for non-sterile compounding. That other portion is for sterile compounding. This area within the pharmacy is for dispensing. This is where we store commodities. So there has to be a dedicated area for non-sterile compounding. Why do we need a dedicated area? Because it will help prevent cross-contamination from other pharmacy operations. For example, if you are dispensing some things, you don't want to have what you're dispensing mistakenly appear on the table where you're doing your non-sterile compounding, so that you don't mistakenly think it's part of the ingredients you are putting into the compounding, the medication you're compounding. You also don't want to do it within a storage area. You just want to ensure that there's no, cross contamination of anything happening. That's why you need a dedicated area, and it should be physically separated from, areas handling hazardous substances. In pharmacy, there are products we call poisons, not because they will kill you, but because there are special products that should be only received by prescription because of how potent they are. If you mistake 0.01 or zero point 0.1, it can be harmful. So you have to ensure that your non-sterile compounding, because you don't take all the safety precautions you take in the sterile one, where it has to be secluded and everything. So you have to just make sure that there's an area that is dedicated for non-sterile compounding. Let's just not say you can compound anywhere. It's easy for you to want to just pick what you want to compound, put in a mortar, and be walking around and compounding. That's not best practice. We're saying you need to have a dedicated area. That's bullet number one. Talking about facilities and environment for non-sterile product compounding. The second point is you want to make sure that the place is clean and well ventilated. The workspace is very clean and well ventilated. Anything medication, you have to think safety first in preparation, in administration, and use all the time. Ventilation, why? Ventilation will prevent, accumulation of dust, airborne contaminants, or vapors or anything. So if you have the area very well ventilated, you're sure that you're working without dust. When there's no ventilation, dust can easily come and settle, and that will contaminate your preparations. So we want to ensure that the place is clean and very well ventilated wherever you will carry out your compounding. Cleanliness especially minimize, how, bacterial growth and product contamination. If you don't, microorganisms are there the moment you allow them settle in a particular place, depending on the temperature, humidity. Some are very fast. They grow, before they grow very fast and contaminate. So you have to be sure that the place is dry before you start your compounding. The place is ventilated and quite clean, so that you don't encourage the growth of microorganisms. Your floors and workbenches should be easy to clean and disinfect. So you use such, materials for your work floor or benches to be easy to clean. If you go to most pharmacy stores or spaces these days, you find that the kind of floor they encourage is epoxy floors, something that you can easily wipe and it will just go away, instead of some other kind of floors that will be difficult to clean and then help bacteria to grow and contaminate your area. So that's point number two, really very important. And then adequate lighting and temperature control. Like you all know, in compounding, we'll get into that as we go further down. You have to do a lot of measuring. It could be liquid measurement, it could be powder you're measuring, a lot of measurement. So if you don't have enough light and you're compounding, you could miss that weight that you're required to get, or even take the wrong thing if you're mixing, if you don't already have them on the workspace. So lighting is really very important. Adequate lighting, of course, not too much light, but adequate one that will be adequate for you to see, and ensure that whatever you're inspecting before you use, you're seeing it very well. Whatever you're measuring, you're getting the measurement very well, and in weighing them and all that. You do know that temperature and humidity must be controlled. I mentioned that earlier. Must be controlled to prevent, risk of microbial organisms growing in the course of you. You don't underestimate how fast it can take to have the right temperature and humidity for microorganisms to begin to grow and contaminate your work. And yeah, temperature control help stability. You also know that as we go further down, we'll discuss a lot of substances are temperature sensitive. So we'll have to ensure that we're controlling the environment for temperature and humidity, depending on what you're compounding or bringing together. You have to be sure you're doing that under the right temperature and humidity. Remember the pharmacopoeia I mentioned, the official document for standards for every preparation of every pharmaceutical preparation. There's British pharmacopoeia, United States pharmacopoeia. There's standard for everything, so you know the right temperature and humidity you need to have for every compounding or preparation that you need to make, and make sure that you have that temperature before you continue. In terms of access, it is encouraged that we restrict access to reduce contamination. Only authorized personnel should go in and out to avoid contamination, so that you are sure that authorized personnel would know that this is what is required in terms of how the hygiene you're supposed to keep. Wash your hands before you enter, or clean with a towel and all of that. So non-authorized personnel should not be allowed into your compounding space, and prevent unnecessary movement and introduction of contaminants. Even you yourself, the person compounding, prevent unnecessary movement. Don't keep your phone somewhere. As you're compounding, you're going to pick it and see who is sending you WhatsApp chats and dropping and coming back and using the same. And your phone may just contaminate your hand, and your hand contaminate your preparation. So such things, we need to actually watch out for and be sure that we are not contaminating work compounding that we're doing. And then proper storage for ingredients and products. The raw materials, you need raw materials to do any compounding. The raw materials, even the finished products, must be stored according to manufacturer's requirement. Again, it must be stored according to right temperature. Some of them, they will say don't put in the fridge. Some of them you have to refrigerate. Light, moisture, environment, you just have to have the right conditions for storage. The raw materials and the finished product, you must make sure you're storing them at the right. That's why the pharmacopoeia, like I mentioned the other day, the British pharmacopoeia or US pharmacopoeia. I know Nigeria has one, British Nigerian formulary or something like that, but the standard, the global standard is either British pharmacopoeia or United States pharmacopoeia, and this is like the Bible or the Quran of pharmacy work anywhere. You have to consult that to ensure that knowing the temperature. No, this preparation is for the eyes, so let me keep it in the fridge. Or no, this preparation is topical, it's for the skin, we don't need to put it in the fridge and all of that. You need to consult that book to be sure. Clear labeling, make sure that the labeling is very, very clear, and separating ingredients into, as we'll see in the next discussion, down the slides, we have active ingredients, excipients, and other materials. Just separate them clearly. This one is active ingredient. Don't mix them with excipients. Don't mix them with preservatives. Don't mix them with hazardous materials. Don't mix them with the vehicles. Just make sure you're arranging everything accordingly. And most importantly, in your compounding area, you're identifying expired or damaged stock to be put in one area. In pharmacy, we call it quarantine. Quarantine them in one area and make sure that they are, away from where anybody coming to compound next would pick them and use them. So this is a rundown of the facilities and environment required for non-sterile product compounding. I'm deliberate in making these repetitions that I'm doing, because remember in the last class, I said we're going to be doing low dose, high frequency. We're not going to take too much every week. We'll just take a little and then we repeat them all over again so that we are able to internalize them. And I know that when you go for the internship, you will suddenly remember that we discussed all of this. We'll have the notes anyway. When we submit all of the updated slides, we'll have the notes. So the picture you're seeing here is, like I explained, these are modern ways to do non-sterile compounding. So you see that the pharmacy area will have that modular, section where it's naturally regarded to be clean. So when you want to do compounding, you go into that area that you see secluded, different from the compound dispensing area where everybody comes over the window and you're able to exchange, commodities and all of that. But you have that in modern days, but in our time, to be honest, it's just a big room with several benches, and we know that this area is for sterile compounding, secluded, but the other benches are open, just open air. But we just make sure that the benches are clean and you know where everything is stored. But modern day, this is what you see. Ventilation and air quality system, again, during our time, you make sure you have the windows very open, and then there's adequate ventilation, everywhere is dry. But these days you now have, the ventilators, ventilation and air quality systems controlled, for you to do your work. There are, machines, called compounding aseptic contaminator isolators also. You want to compound, you can, use such specific areas, depending on how sensitive even the non-sterile compounding is. And then, storage areas, everything well labeled and very well sectioned. I want to stop to see if you have questions or you want me to continue. Run down and then we can take questions later. Okay, next slide. Right. So our next slide is actually, like I mentioned, equipment and material. We finished talking about facilities and environment. We're now talking about equipment and materials. Equipment and materials for non-sterile compounding, non-sterile products compounding. So generally, you will find the use of all of these categories: measuring apparatus or tools, mixing tools, balancing tools, packaging, and PPEs. And we'll take them one after the other. So in terms of measuring, you can see in the picture there, measurement, of course, when you say measurement in compounding, normally it's to do with some liquids or preparation. Of course, you measure solid things, and in pharmaceutical lingo, it's better you use balancing for such. It's better you use balancing for weighing and measuring things that are in solid form. But in terms of liquid preparation, so you need graduated cylinders like the one you're seeing there. There's a graduated cylinder you're seeing on the screen there. You're seeing also, syringes and pipettes. These are the things you commonly find within the non-sterile compounding table for measurement. You want to take volumes, take measurements. For where you have to pour 100 mils, 20 mils, you use the big graduated cylinder. But where you need to put something in a very, 0.1 or 1, drop or something, you use syringes and even pipettes. Pipettes are the ones that will measure to very minute, pipettes have this pressure function where you put them in the end, as you remove your finger, the thing sucks up. So pipettes measure very, very minute distance. So in terms of measurements, You use the graduated cylinders, syringes, and pipettes. They provide, you your accuracy in measuring liquids mostly. Of course, like I mentioned, smallest used for syringes for smaller things and pipettes to measure very, very small things. So in terms of liquid measurements, these are things you commonly find in an area where you do non-sterile compounding. Now, the next thing is mixing. And you can see mixing is important. In fact, the word compounding is actually bringing a number of things together into one particular place and making them stay together as one. So mixing, you really need mortar and pestle. That's the mortar you're seeing with the pestle in it. You need a stirring rod or homogenizer. So for mortar and pestle, is something you use to make sure that you're breaking the solids that are big into smaller, finer, powdery form. In pharmacy, it is called titration. When you have a big solid that you want to crush into smaller, finer form, is called titration. Trituration, spelled Trituration, trituration. So you triturate. That's the process of using that to crush big things as triturate. And then for stirring rods, there will be a time you need to want to mix liquids. You're bringing two different liquids, you're putting them together, you want them to be together, so you use stirring rods to together. These are things that you should remember, um, because when you go for internship, you'll come across all of them. You'll have the notes, and hopefully you'll be able to remember that these things were mentioned and you're able to use them. There are even homogenizers. Homogenizers are also some instruments you use to ensure that you know you're making, when you want to disperse something in a uniform fashion. For example, let's take emulsion. Emulsion is anything that has powder and some syrups that you want to mix together. You don't want to mix it in a way that will leave the powder or semi-solid in one portion and more of the syrup in one portion. So you want to ensure that everything is evenly distributed. The powdery ingredient is easily uniformly distributed. So you use a homogenizer. The instruments exist for that. Yeah, so that's in terms of mixing. You need all of that. Let's not forget, was where we talked about measuring devices. You need to measure and then you mix. These are even the steps following. You measure, you mix, and all of that in compounding. The next one we're going to talking about is balancing. So I talked about measurement in the sense of everything is about context, right? So we talked about measurement here in this pharmaceutical space in the context of liquids. Now, the same measurement in the context of solids or powder is you use what is called a balance. It's called balancing. Why is it called balancing? In those days when they started trying to use balance to measure powder and know the quantity you need to use for a particular compounding, is like a scale, a scale that has two parts. So you put something that you know the weight on one point, and then you keep adding powder or the active ingredient you want onto the other part until there's a balance. Then you now say, okay, since I know the weight of this one to be 5 milligram, I have achieved 5 milligrams. So that's why it's called a balance. And there's a reason why in pharmacy it is deliberately, not say we're all measuring liquids and solids. It's just so that we have this differentiation that when we say balancing, we're talking about measurement and weighing things that are in powdery form or in solid form. But when we say measuring, we're using cylinders and all of that to measure. Generally, there are two main types of balances. Let me say there are two main types of balances. You could have an electronic balance and a torsion balance. Torsion balance is the one that I explained earlier, where you have two sides, and then when you put something on this, it goes down and raises this one. and then you put some weight here to be able to balance and say, okay, now I know I have 1 milligram since the weight of this one has been determined to be 1 milligram. That's torsion. That's the torsion one. So with that, with advancement and civilization, we now have electronic balances, the ones that you plug to a source of electricity. You already calibrate it, plug to a source of electricity. Just take that powder, put on a particular tray. I think if you if you go to the next slide, uh, IT, if you go to the next slide, there's an example of a balance, an electronic balance. If you just flip to the next slide and we can come back to this. Yes, so are you seeing that blue container, blue bottle is right in front of a balance? That's an electronic balance. So you put whatever you want to weigh in that dish and then plug it to a source of electricity or battery. And as soon as you put, it's telling you this is 2 milligram. On that screen there, it will show you 2 milligram or 10 milligram. So this is an example of an electronic balance. And these electronic balances actually do measure to a very minute quantity. If you want to measure 0.1 milligram of something, this will help you. But it will be difficult for you to do that with the torsion balance because you need to find something that you put on this side that will balance that. So let's go back to our previous slides, please. So right, so these are things that you will naturally find in a particular compounding room for any pharmaceutical non-sterile compounding. Yeah, measuring, mixing, balancing, and then now packaging. You definitely is really important in non-sterile compounding. You find packaging materials. You have vials, you have bottles, you have jars and syringes. Some preparations you do, you save them in syringes and then they are for immediate use. Some preparations are put in vials. Vials are these small containers, bottle-like containers. And then bottles are the bottles, for syrups and cough syrup and all of the bottles. And then jars, depending on the size of the preparation, you can have jars. You have generally packaging material. You even have cartons, small, containers where you put, sometimes you have these dispensing envelopes, they call them, that could be nylon in form or even paper. So different packaging materials you find within the non-sterile compounding space. And then lastly, PPE, personal protective equipment, gloves, masks, lab coats. This is easy to make you visualize. If you remember the COVID era, those people that the public health personnel that were intervening in COVID, they were responsible for taking blood samples, and you remember how they are dressed from head to toe, covered with space to see, the eyes, a transparent portion. So that's personal protective equipment. So it doesn't have to be complete like that of COVID. Depending on, what preparation you're making, sometimes it's important you cover your hair with something. So, cover your hair, put gloves, this for non-sterile compounding, it's important. You don't have to wear all that, but you have your lab coat, as a pharmacy technician, every time you go into a place where you even dispense medications, you need to have that lab coat for ease of recognition and also so that you're protecting yourself. So lab coats, masks, gloves, all those personal protective equipments are really important. Yeah, I see time is fast spent. So that's equipment and materials, equipment and materials. Yeah. Any questions, or do I still go ahead? We end the slides and then we're able to interact at the end. If that's consensus, let's move on, IT. I hope I'm not too fast today.
No, please, you're perfect. Thank you.
All right. Thank you. Thank you, Dr. Stephanie. All right. Let's go to the next slide. I mean, these are things we all talked about. Right. So this is where I actually wanted to spend some more time. I see that time is not our friend today. And these are compounding ingredients. And if you don't take anything away from today's class, please remember these three classes of ingredients that we have here. If you don't take anything at all today, if you come for next week and I'm asking for a recap, if you don't remember anything, please remember these compounding ingredients that in any pharmaceutical preparation that you know, in any pharmaceutical, as long as it carries the name pharmaceutical and it's a preparation, you must have active ingredients that are called active pharmaceutical ingredients, APIs. Not the APIs, I'm sure IT is listening to me now, not the APIs of IT, where you have some, APIs that you need to link to whatever, These are active pharmaceutical ingredients. In every pharmaceutical preparation, there must be active pharmaceutical ingredient. If you have a preparation that is given to you as a pharmaceutical preparation that does not have an active pharmaceutical ingredient, it's called a placebo. So it actually exists where you can have the preparation, it's given to you as a medication, but it lacks the active pharmaceutical ingredient that's supposed to cause that pharmacological effect. It's called a placebo. It's used in such things as psychiatry, yeah, where you treat psychiatric patients with a lot of drugs. You think they're getting better, they are dependent on it. When they see you, they want you to give them the medication for them to feel all right, but you know they're getting better, they don't need it, so you give them placebo without the active pharmaceutical. So it's the same medication prepared just without the active pharmaceutical ingredient. He takes it, he feels, yeah, I'm fine, because there's also something called psychotherapy in in psychiatry. Psychotherapy is when you use psychology to treat someone. You're giving him something is empty, but you're telling him just take it, you'll get better, and the person believes you and then it works for them. So again, I'm taking some time to explain that for every preparation that is called a medicine or a drug, a pharmaceutical product, it must have an active pharmaceutical ingredient. It must have what is called excipients. Now, excipients are three different things. And you could find all at the same time in the preparation. You could find some without the other. So excipients include diluents, and in explaining what a diluent is, let me take some time to. So that was why I wanted to take some time. So I believe that we are okay with the active pharmaceutical ingredient. Is clear that is that particular compound, normally in small proportion compared to excipients, is normally a smaller quantity to the excipient. I'll give you an example of paracetamol. In paracetamol, when they label it, you see paracetamol 500 milligram. Now, the active ingredient is actually 500 milligram, but if you weigh the entire thing, maybe it's the other portion will be starch, while the active ingredient is just something small within the starch. And so when you take that tablet, you're not only taking the active ingredient, you're also taking starch, you're also taking some stabilizers, some preservatives, all within that tablet that you take. So that active pharmaceutical ingredient is the thing that causes that pharmacological effect. You must source it from a good supplier, and there's something called a certificate of analysis. For if you are the one procuring, you have to make sure it comes with a certificate of analysis for every active pharmaceutical ingredient. It's been analyzed to cause this effect, this effect. So that certificate of analysis must accompany it. Now, excipients, let me take some time to say there are three things, like I mentioned: diluents, stabilizers, and preservatives. These three things combine help improve stability of a medication, for it to be stable, not to disintegrate the moment it comes across light or something that is not part of it. And then for palatability, to make it palatable, and then just generally usable. So these three things are called excipients. Excipients, if you put them together, are called excipients. So what are diluents? Diluents are that bulk forming agent. Like I explained, paracetamol is actually acetaminophen in small quantity, put together and compounded in that bulk, it could be starch, it could be lactose, it could be microcrystalline cellulose, So the point is that that active ingredient must stay within something that is easy to take, is safe to take. So mostly starch and lactose, and cellulose as well. So that those ones are called the diluents because kind of you collect the acetaminophen, dilute it to this, and then it becomes paracetamol. Then you also add stabilizers, like I said, things that will help them not to degrade the moment they come in contact with light. Things like antioxidants and other, names that, let me not just start mentioning sodium, metabisulfate, and all of that. So these are stabilizers. You put them in medications to stabilize the medication, to fight against degradation, to stay that shelf life that you have expiry date in 2 years or 1 year. They help stabilize. And then you have preservatives. These are the things you put to also help you inhibit the growth of microbial organisms so that, they're in there. If any microbe or anything, you open, you take the syrup, you close. If you introduce anything that should cause growth of, and these things will help you. Common one, is parabens, and other some chlorides like that. So these three things make up excipient, and the last one is vehicle. The vehicle is actually the medium that carries all of this. So assuming we're doing a syrup for a child or a suspension for a child, you would have the active ingredient I mentioned, acetaminophen, if we're preparing paracetamol suspension for a child, acetaminophen, and then you have that starch that will carry it, and then you have a vehicle. It's either water, syrup, glycerin, ointment bases, creams, gels, there are numerous there. So you have to have something that will carry them. So again, I'm saying in terms of compounding ingredients, non-stellar compounding ingredients, do not forget, we have the active pharmaceutical ingredients, we have the excipients made up of diluents, stabilizers, and preservatives, and then lastly, we have a vehicle that will have to carry all of what we're preparing. And of course, the vehicle must be compatible with the active pharmaceutical ingredient. So the choice of whether I use a purified water or I use a glycerin or I use aloe vera syrup or I use whatever will depend on whether that active pharmaceutical ingredient will be compatible with this, will not just degrade or fight the vehicle. And again, all of those are found in the pharmacopoeia. All the standards, what will be compatible and not compatible, you find them in the pharmacopoeia. Now, I won't have time to talk about the pharmacopoeia today, so maybe we'll continue pharmacopoeia next week because I want to give time for us in this 30 minutes to have questions and discuss. But IT, just show the next slide, which should be a picture of pharmacopoeia. So these are huge documents. Like I said, the official documents that have for every medication on this earth, you find their preparation here, except they are done behind the scenes. But the ones that are official, approved by NAFDAC or other drug regulatory agencies in other countries, you have to find them here. What is a paracetamol? What is the active ingredient? What are the excipients? What are the things you can use in what quantities? And all of that. So I'll talk about this next week because of time. I'll reserve these 12 minutes for us to now, take questions and, clarify a few things before we close. So let me stop talking and ask for questions. If you have any questions or clarifications requiring, me to respond. I'm not the only pharmacist. Yeah, I see pharmacist Blessing also, Dr. Blessing. So if there's any need to respond to such things. So 11 more minutes, questions and answer before we close. There are no questions. It's either you understand perfectly or completely have lost everyone. Questions? You want me to call names? Any questions? Please ask questions. Or is everything crystal clear? Otherwise, ask questions, give comments, anything. Yeah. Yes, Arize, please go ahead. Arize, right? We can't hear you if you're speaking. We can't hear you if you're speaking. Can anyone hear him? Please, can anyone hear Arize?
No, I'm not hearing.
Okay. Arize, we can't hear you. Maybe something wrong with your mic, Arize. If you can unmute and try again. In the meantime, anyone with any questions can bring them up. Any
So,
questions?
so
Yes, carry on, Doc.
good evening, Dr. Yakubu.
Good
And so,
evening.
yeah, so my question is not just for me. It's
Yeah,
perhaps it could be of value to other colleagues and students on the call. So
absolutely.
I would imagine that compounding itself as as a component of any pharmaceutical endeavor, right? Pharmacy is as old as pharmacy itself, right? Is as old as
Correct.
medicine
Correct.
or any therapeutic you know
Yeah. Process.
evolution in
Correct.
history.
Mhm. Correct.
So and you mentioned, surprisingly, my question is on what I'm seeing on the screen right now on
Yeah.
pharmacopoeia, even though I know you
Yes.
haven't discussed it yet. So,
Yes.
and you did mention that pharmacopoeias are standards, right? Either
Yes. Yes.
global standards by which
Yes.
I can imagine that in terms of measurement for
Yes.
paracetamol, it would be in a
Yes.
pharmacopoeia. For the
Absolutely.
adult, for the child,
Correct.
it's going to be a syrup or an injection. The standard measurement
Correct.
will be in a
Correct.
pharmacopoeia.
Yes.
And I'm seeing this pharmacopoeia here are in 2018 and
Correct.
2019.
Correct.
Um,
Correct.
what does that mean? Is it that this is the first version, the latest version? I just got curious and I said, let
Good.
me ask. Okay.
Very, very good question. And I know you know the answer. So you're asking for, you know what they say, I'm asking for someone. So let me say, for example, the British Pharmacopoeia was first published in 1864. And since then, every other year or 2 years, there is a review of the pharmacopoeia. So depending on when it was reviewed, remember they discovered some more medication. Malaria treatment used to be chloroquine, we all know, so it was there. Later, we discovered that, oh, chloroquine alone isn't solving the problem of malaria. You now have to have an artemisinin combination with a lumefantrine. It was discovered Artemisia in China. So, they did all the clinical trials and it passed. They will review the pharmacopoeia and add all that is required. So these ones you're seeing are 2018, 2019. As at 2018, after reviewing and adding or deleting things that no longer are there because some medications go off the shelf, like chloramphenicol, they no longer want to use them anymore, because of some discoveries of some erosion of cartilage and some other things. So you would, the point is, the first version for British Pharmacopoeia, for example, was in 1864. So you can imagine that from that time till now, it's volumes and volumes. The US Pharmacopoeia was actually in 1820, first published in 1820. So I reckon that it's not every year because sometimes clinical trials take 5 years or more. So it's not every year you have to review the pharmacopoeia. But if you review a pharmacopoeia in 2025 and add anything or remove anything, the version will now be 2025 version. So that's it. It keeps, it just keeps evolving, like you said, and things keep making it to the list. and all of that. And because of size, you will see that there are numbers, USP41, NF36. So because of size, you can imagine that if you have one single document that will carry the medications from 1820 or 1864 till now, you'll be, taller than a five-story building or something. So they keep putting them in volumes. And so sometimes if you're trying to look for something, they may refer you to go to USP 41 or go to BP, so number, and then you're able to find them. So that's the answer to that. Yeah.
So, sir,
Over.
as old as these pharmacopoeias have been, that's as old as the practice of compounding. It has evolved over these
Over
years.
time.
The
Absolutely.
practices, the quality, the innovations that have happened over
Yes.
this time.
Over
It's
time.
like,
Correct.
all right. Thank you, sir.
Correct.
Okay.
In fact, even older, the practice of pharmacy would definitely be older than when the pharmacopoeia started. People were compounding and doing other such things. It was in 1864 they decided, hey, let's begin to document these things before we all forget and all of that, what we've done. 1820 in the US and all of that. So pharmacy practice go way back. They were doing things. Somebody would just go to the bush, plug this, plug that, mix them together. Oh no, this your eye, you have eye problem. This leaf looks like the shape of an eye. I'll plug it, and this one also looks like the shape of something. Let me mix them together. But they started documenting them in 1820 and 1864. So you're correct. You're right on point.
Thank
Thank
you.
you very much for that question. Like I said, I know you were not asking for yourself. Any other question? Ariza, are you back? Can we take your question in the next 3 minutes?
Sir,
Otherwise, yeah, please.
sorry, sir, I have no question, sir.
Okay. All right. Thank you.
Thank you, sir.
Thank you.
Thank you, sir. Thank you, sir.
Okay. Right. So next class, we'll start with a recap. And following the recap, we will continue from a copia and, just carry on.